Hello, my name is Ignacio Melero. I work at the University of Navarra Clinic. I'm responsible there for the immunotherapy program, both in research and in clinical practice. My job here today is to address biomarker discovery efforts in immunotherapy of cancer, a very challenging task. And also I will be covering immunotherapy of renal cell carcinoma. These are my disclosures. And actually there are many things you can measure in a cancer patient trying to address whether the patient is going to benefit from treatment. In a way, the simplest dichotomy would be things that you can measure in the patient and things that you can measure in the tumor. And we have many tools. We can go to DNA and genomics. We can look at the messenger RNAs that are expressed. Actually, we can look at the microbiome, not forgetting that the immune system is highly regulated by the flora, the microbial flora. We can check parameters of immune function, both in peripheral tissues or in the tumor itself. We can use several tools of proteomics, and we can look at the epigenetics of the tumor, and all of those factors are going to really affect whether immunotherapy works or not. The problem is that this makes it very complex, and we certainly want to know what are the most predictive factors. Probably more than one factor affects a lot. This is multifactorial. So maybe we will do our best to assess the propensity to respond to immunotherapy by measuring several parameters. There is a review by Christian Blank in Science that I do recommend to address this multifactorial scenario. And in a way, they use a kind of a geometric figure whose area is going to reflect the likelihood of response. And several factors are plotted around this immunogram, as he referred to it. And one of the factors is tumor foreignness. We will go through it. The other one is whether the tumor is already infiltrated by immune system cells and what's the kind of immune cells that are there. Then the presence of the targets for the immune therapy treatments and the presence of the so-called checkpoints in the tumor. But there are other factors such as the microbiome. Then there will be metabolic factors and also inflammatory mediators that we would like to measure. As I am mentioning, this is getting more and more complicated, but for clinical practice we need to make it simple. And simplification is often an oversimplification, but never forget that as it goes in the proverb, simplicity is the ultimate sophistication of complexity. And actually the role of many investigators now is to really identify what are the meaningful factors. So the first one I'm going to refer about is tumor foreignness, defined as the mechanism that provide the immune system with a target to identify the tumor and distinguish it from normal tissues. T cells recognize antigens in the form of peptides presented by HLA antigen-presenting molecules. For HLA class I molecules, this presentation happens because of peptides coming from the transcriptome of the cell are loaded onto the binding cleft, peptide binding cleft of HLA class I molecules. If you look at this representation of the molecule from the top of the cell, you can see the peptide in red and the antigen-presenting molecule depicted in white and blue. And how do these peptides come onto these antigen-presenting molecules? Well, it's kind of a sophisticated pathway. The idea, and probably you know very well, is that the proteasome is constantly degrading misfolded proteins in the cytosol, and it breaks the proteins into peptides of different sizes, some of which are pumped into the endoplasmic reticulum by specific transporters. And once there, with a system of chaperones, they are loaded on this binding cleft of a nascent HLA class 1 molecule, which is mature in the Golgi and eventually comes to the cell surface, where it's presenting kind of the whole array of peptides fitting this class 1 antigen-presenting molecule. And for evolutionary reasons, this locus, which is expressed in a codominant fashion, is highly polymorphic. By that I mean that many different alleles can occupy each of the loci, that in the case of HLA class 1 molecules that present antigens to cytotoxic T lymphocytes are the locus HLA-A, HLA-B, and HLA-C. And as you can see, they can be occupied by quite a few different alleles. The reason for such a diversity is that they have evolved in humans and in apes in order to accommodate the most common pathogens, and there is a survival advantage if you can present dominant antigens from prevalent pathogens. But this complicates a lot the life of transplanters, but also of tumor immunology. An important thing to bear in mind is that if you look at the binding cleft of HLA class 1 molecules, there are several areas that are fitting amino acids of the peptides. Those are the so-called pockets that accommodate the lateral chains of some of the amino acids in the peptide. And because of that, there are patterns of peptides that can accommodate this binding cleft. These are usually, depending on the HLA molecule, 8 to 9 amino acid long peptides. And interestingly enough, when proteomic people started to elute these peptides and sequence them, they realized that there were shared patterns in such a way that suggested that you could make predictions of which peptides could be eventually accommodate a class 1 molecule. As you can see here in the picture, there are these anchoring amino acids represented in orange that are actually common in the many different peptides that are eluted from a given HLA class 1 molecule. And tumors, to be tumors, need to have mutations. And mutations would potentially give rise to antigens, to antigenic determinants presented by HLA class 1 or class 2 molecules. Whether point mutations that are non-synonym in terms of protein sequence, deletions, insertions, would give rise to sequences that are not present in the normal transcriptome of the cell, and thereby can give rise to peptides, but not antigenic peptides, but not always, only when they do fit the endogenous repertoire of antigen-presenting molecules. Problems with RNA splicing or even epigenetic problems can eventually give rise to potential antigens. Ooh. So whenever a mutation happens, it can be antigenic for a number of reasons. One of them is that it will result in an amino acid that is not present in the native sequence, that it's phasing out, and it can be recognized by the antigen T-cell, the T-cell antigen receptor. Differently, it can also generate an amino acid or a set of amino acids that would fit better with the antigen-presenting molecule, thereby giving rise to a neoepitope or a neoantigen. And then if the mutation is more complex, several of these things can happen together. But those are the basis for antigenicity in tumors. The interesting thing is by the group of Tim Chang in the Memorial Sloan Kettering Cancer Institute. it became clear that the more mutations a tumor had, in this case in metastatic melanoma, the more likelihood to clinically benefit from a checkpoint inhibitor, in this case blocking CTLA-4 with the antibody ipilimumab. But not only mutation burden. If now they apply algorithms to predict the likelihood of these mutations to fit with the HLA alleles present in the genome of the patient, then the correlation with survival became even more clear. So it's possible that the main factor linking antigenicity with tumor burden and prognosis or predictive value for immunotherapy is related to the number of epitopes that are actually, the neoepitopes that are actually presented by tumor cells. This seems also to be true for non-small cell lung cancer. The pioneer group work by Dr. Ritzby in this regard showed that there was a correlation of the clinical benefit from pembrolizumab in these patients depending on the mutation burden. And again, when neoepitopes were predicted in silico, there was a good correlation between overall progression-free survival in this case and of the patients following pembrolizumab treatment. Interestingly enough, using the tricks that immunologists use to actually be able to see that the patient has T cells responding to these antigens, it was possible to see an increase in the immune response as a result of PD-1 blockade, and it was possible to see that some of the epitopes neoepitopes were actually recognized in the patient, because if you got those T cells and exposed them to synthetic versions of the antigen, they started to produce gamma interferon that can be measured by intracellular staining, and that's what these dot plots here show, is that some of these antigens were actually recognized by larger numbers of T cells in the patients. Another important point coming from the Cree Institute here in London in this paper by McGranahan et al. Published in Science is that they used a trick to actually be able to tell whether the mutation was present in all the tumor cells or it was heterogeneous. Is what in tumor genetics they call an stem mutation, which is there probably all the way from carcinogenesis, or what is a branch mutation that is not shared by all the tumor cells. And as you would probably have predicted, those patients in which the neoantigens are in the stem, are in all the tumor cells, are more susceptible to immunotherapy than in those cases where there is multiple clones of tumor cells expressing different mutations. And this is probably due to the fact that all the tumor cells are expressing on all the antigens, which are ultimately the target of the immune response that we exploit in cancer immunotherapy. Indeed, using these algorithms to predict heterogeneity in the expression of antigens, they could see a nice correlations to tumors. These are not easy biomarkers at all, because they presume that you have to do whole exome sequencing of the tumor, normal tissue, typically peripheral blood, compare them, and then do a lot of bioinformatics in order to predict the epitopes. But if it were a good biomarker, eventually costs of sequencing and of bioinformatics can be dropped, and this could be done in the future. Another very important point, and this was realized by Tom Gajewski in the University of Chicago, and also by Franco Marincola at the NCI, as that the tumors that tend to respond to any kind of immunotherapy, whether vaccination, immunomodulation, whatever, tend to be those that have already an infiltration of T cells in the tumor, in such a way that immune infiltration in itself is very often a prognostic marker, a favorable prognostic marker in tumors. But, and this is an interesting trick, what probably immunotherapy does is to unleash an immune response that is already ongoing. And unless you have an immune response already ongoing, then immunotherapy tends to be not successful. In this way, we can classify tumors as hot and as cold. Those tumors that we call cold tumors are tumors that do not have T cells inside. Then we have tumors with evidence for pre-existing immunity, particularly those with a lot of CD8 cells or T cells producing interferon gamma. Those are what we call hot tumors. And then obviously you have a spectrum where some of them are colder than others. And there is something also that we see a lot is what we call the excluded infiltrate, and those are tumors with an invasive margin with T cells, while the malignant tissue itself, where the tumor cells nest, is devoid of T cells. And what defines a hot and a cold tumor? It's very complex. Actually, a lot of the research right now in cancer immunology is trying to address precisely this point, because there are factors that are related to the genetics of the tumor, to the epigenetic of the tumors, with the kind of inflammatory response that it's there. And actually it would be kind of very difficult to generate a very good algorithm to predict these. Things are getting very complicated, as you can see here, and if you consider all the functions that you need for a successful immune response, virtually you could be measuring all of them and trying to figure out whether or not this is giving rise to an effect on immunotherapy. But at the end of the day, what has already been successful is that we know that checking the transcriptomic signature of genes that respond to interferon gamma correlates with propensity of response. There are several signatures that have been validated and patented, and they are routinely used. This is simple because it's a simple macroarray that can be done. Then the evidence for activated CD8 T cells, if they have the molecular means that they use to kill, such as perforin or granzyme B, and then the type of macrophages that are inflammatory and are producing a kind of the right type of mediators, such as TNF-alpha. Those are what we call M1 macrophages, and they are typically elicited also by interferon gamma, and they are nice guys for immunotherapy, as opposed to what we call M2 macrophages, which are producing angiogenic factors and growth factors, and the kind of inflammation driven by IL-6, for example, that it's not a myeloid inflammation, that it's not good for immunotherapy. Obviously, in a population of cancer patients, we get these parameters in different fashion. It could have kind of a bell-shaped curve, and some of them are more propense to immunity, some of them more to tolerance. Genetics, age, the microbiome, the presence of previous viral infections, the exposure to carcinogens, the exposure to all that defines this propensity, and then it's very multifactorial. But the methods that we typically use to analyze all these are conventional immunohistochemistry. We are learning now that it's much better to have more than one parameter information in the tissue section. And for that reason, multiplex immunofluorescence techniques are becoming widely used, and actually they provide a lot of information. And since fluorescence is better than chemistry with regard to quantitative measurements, immunofluorescence can provide semi-quantitative assessment of the level of expression of the different proteins. Using RNA-seq or different forms of RNA expression techniques of multiple genes in a single run are also very important. And as I mentioned, and mainly because of analyzing the foreignness of the tumor, the number of mutations, and their likelihood to give rise to neoepitopes, we use whole exome sequencing. And we are trying to learn also whether there are genetic or epigenetic features of the tumors that are mediating this immune exclusion, something that has a lot to do with the vascularization of the tumor and the type of microvasculature that it's in the tumor. A very interesting suggestion came again also from the group of Tom Gajewski, and that comes from the fact that cytotoxic T lymphocytes, CD8 T cells, typically to get them turned on, you need to present antigen with a very interesting subset of dendritic cells, of antigen presenting cells, or professional antigen presenting cells. This small subset is characterized by expressing the transcription factors BAT3 and also IRF8. And in a way, what we have learned more recently is that at least in melanoma, but probably also in many cases in the TCGA, it comes a very interesting feature is that hot tumors tend to be richer in this particular subset of CD103 dendritic cells than those who are not. And even then, you could see hot tumors in red, cold tumors in blue here, and you set your cutoff in the middle. And it's interesting because this correlates better with the presence of these professional antigen-presenting cells for CD8 cells than actually the mutation burden that is represented here in the upper graph. So it's probably the case that it's not only gene mutations what make tumors recognizable by the immune system, but also, and importantly, the presence in the tumor of these antigen-presenting cells. And actually, those tumors which are immune deserts that are not populated by T cells are probably primarily devoid of these antigen-presenting cells in the infiltrate. We don't know what's first, the chicken or the egg, but this is a field that is going to be very active in the next 5 years. An important practical knowledge is that B7H1 is another name for PD-L1, and the presence of TILs can be combined together. And by the different combinations, you can get 4 phenotypes, 1, 2, 3, and 4 here. Some of them which are deserts, do not have PD-L1 nor tumor-infiltrating lymphocytes. Some that have PD-L1 but not TILs. Those that have both of them expressed, and some of those which only have TILs but not PD-L1. And this is probably a very important thing because we can personalize treatment for each of these subsets of patients. Those who have an immune response going on and PD-L1 is there probably blocking the response are going to be those patients who are more likely to respond, as opposed to the immune deserts where probably we will have to do something else in order to get an immune response against the tumor, something like vaccination or a co-treatment that would enhance this infiltration by immune system cells. So I guess that now you are going to watch this video about PD-1 and PD-L1 blockade from a molecular perspective.
In recent years, immune checkpoint blockade has taken centre stage as a significant breakthrough in medical cancer therapy. Activation of T cells involves several paired molecular interactions, such as T cell receptors or CD4 with the major histocompatibility complex, B71 with CD28 and several others. Programmed cell death 1, or PD-1, is a member of the CD28 superfamily and is expressed on T lymphocytes. PD-L1 and PD-L2 are its ligands on cancer cells. Mechanistically, T cells use PD-1 to probe cancer cells for PD-L1 and PD-L2. When they find it, they leave tumour cells alone to survive. When PD-1 binds to its ligand, it induces immune suppression in the tumour microenvironment, which subsequently leads to resistance of cancer cells to the immune system. However, when a molecule blocks this identification, T cells are no longer misled by the presence of PD-L1 and PD-L2, and subsequently rally an immune system attack on the cancer cell. Antibody-based PD-1 or PD-L1 blockade has achieved strong success in clinical trials. These agents take advantage of specific binding to antigens and thus have been widely used for immune checkpoint blockade. Multiple PD-1 or PD-L1 antibodies have been approved for clinical use or have entered into clinical trials, including nivolumab, pembrolizumab, atezolizumab, durvalumab, and avelumab. The first clinical trial of a PD-1 blocking agent began nearly a decade ago in patients with blood cancer. Today, more than a dozen trials have been completed, with more than 50 underway, involving thousands of patients with several different types of cancer. These ongoing trials are encouraging, particularly since they indicate that a substantial number of participants have benefited, resulting in a need for further trials. Additionally, they have suggested that these agents may be effective against multiple types of cancer.
Having seen that video, it becomes important to know whether the tumor is expressing PD-L1, whether there is PD-L1 expression in what we call the tumor microenvironment. And there are several agents blocking either PD-1, nivolumab, pembrolizumab, or PD-L1 such as atezolizumab, durvalumab, or avelumab. And it's not easy at all to detect PD-L1 expression on formalin-fixed and paraffin-embedded tissue. There were many attempts, many cases of failure to generate good antibodies staining this moiety in the tumor tissue. And there are several issues about epitope retrieval, then the folding of the protein, and other factors that makes this very, very difficult. In such a way that the companies developing these compounds have invested heavily in developing or buying reagents that would reliably quantify the number of cells that are expressing PD-L1 above a certain cutoff. These companies have developed their antibodies and automatized the detection and and how you quantitate the number of cells, and you have here several antibodies, 28A8, 22C3, SP142, and SP263. And importantly, since these compounds are being developed in the same period of time, it has become very important to actually be able to compare what's the result with the different antibodies. It is also very difficult to have the pathology departments running all the tests and all the platforms for detection. In most cases, the technique is only taking care of expression with a surface pattern on tumor cells, and then you quantify a brown cell and cells that are not stained, and you end up with a percentage of expression. There's one exception, is that the atezolizumab being developed by Genentech, they were more interested in checking not only the expression on tumor cells, but also on immune cells infiltrating in the tumor, since they found a correlation with the likelihood of response, of objective response to atezolizumab, at least in some types of tumors. In this case, they generated a score depending on expression in tumor and expression on tumor cells. And then they studied different cutoffs in the different studies, using it as a predictive biomarker. As you can see in several attempts to see the consistency of staining with the different compounds, you can see that most of them have a nice correlation. One of the problems is in the in the steepest part of the curve of correlation, where very little differences can mean a lot, and probably is where the cutoffs are. And in the case of SP142, it disclosed less positive cells, but remember that this antibody was generated probably mainly to detect expression on immune cells. This is how the thing looks like. So as you can see, it's not that easy. These are typical examples. So when it is 100% of the tumor cells, it's easy, but then it becomes a little bit less cumbersome, because staining is typically patchy and tends to concur with the areas where T cells are. And one of the reasons for that is that the main inducer of PD-L1 expression is interferon gamma, which is a cytokine produced by tumor-infiltrating lymphocytes if everything is going okay. And then the predictive value as a biomarker after this complex detection in the pathology department is good, but it's not great. By that I mean that you can enrich for patients who would eventually respond to PD-1 or PD-L1 blockade. But as you can see, depending on the disease, this level of enrichment is twofold in the best of the cases. And that's probably not enough to negate treatment to people who have no other treatment with known efficacy. So we really need to get better and beyond PD-L1 staining in the tumors. And again, we go to this nomogram, trying to see what else we can try to plot together in order to have a better picture, knowing that the complexity is great. And there is something which is emerging as very, very interesting, and that's the presence of bugs in the gut, potentially also in the skin. As you know, the microbiome is a huge collection of bacteria that nest in the gut, especially in the colon, and with many species, and that we know for a long time that they regulate immunity quite a lot. In fact, it's species specific as well as a healthy presence of bacteria, giving rise to important components in the pathogenesis of autoimmunity, for example. In fact, the gut microbiome could be not only prognostic or predictive factor for immunotherapy, but also potentially a predictive factor for immune-related toxicity in cancer patients, because there are many mechanisms that are regulated by the presence of some components of the microbiome that go to the level and degree of infiltrate in the tumor and are governed by controlling the function of dendritic cells at this level, but also systemically. If you are curious about this, I do recommend some reviews, including one by Lawrence Bohel and Tom Gajewski, who were the pioneers showing in mice that the microbiota actually regulated the immune response against cancer. And not only the basal immune response, but also, and importantly, the response to PD-1, PD-L1 blockade, or to CTLA-4 blockade. Another important issue is resistance. Immunotherapy is not going to be an exception to other cancer therapies. Tumor cells can develop resistance or be resistant to begin with. Resistance in tumor cells can be present in all the tumor cells in the tumor mass. Maybe some clones will adapt. Maybe at the end of the day, you could have a very, very nice response, but persisting cells would adapt. And the molecular basis for these are only being started to be studied. This is mainly because immunotherapy was not very effective for a number of years. And it is since the first decade of the 21st century that we are achieving some degree of efficacy, mainly in melanoma, that now is extending to other diseases. So in the absence of efficacy, we did not worry that much about acquired resistance. And resistance, and this field has been pioneered by the group of Tony Rivas in UCLA, can be primary depending on genetic and epigenetic features of the tumor. For example, several oncogenes make the tumor less likely to be inflamed, to be hot. But tumors can escape also by downregulating the antigen presentation machinery that I introduced to you at the beginning of the first part of the lecture. For example, downregulating the molecules needed for antigen processing and loading, and importantly, the antigen presenting molecules itself. In fact, HLA class 1 molecules are stabilized. They need to be stabilized by a protein called beta-2 microglobulin, which assembles and stabilizes the complex. And in the absence of beta-2 microglobulin, there's virtually no HLA class 1 molecules on the cell surface. And in that setting, a complete loss of beta-2 microglobulin would give rise to a tumor that's invisible to CD8 T cells and thereby would provide an escape route. HLA class 1 molecules are typically expressed at a very low level. And only when there is type 1 interferon or gamma interferon around, where there is signals that are denoting a viral infection, is when these gets expressed at a logarithmic scale, several logs of increased expression. So precisely this pathway of signaling from the interferon gamma and the type 1 interferon receptors can be down-modulated by mutations or some kind of epigenetic adaptation. And if that happens, there will be a much decreased level of antigen presentation in the tumor. And that's precisely which seems to be a major route of escape precisely in melanoma. The type 1, and in this case the interferon gamma receptor, relays on two tyrosine kinases that get associated to the cytoplasmic tail, JAK1 and JAK2, that activate the STAT1 and STAT2 transcription factor that relocates to the nucleus after phosphorylation and regulates many genes, including, importantly, HLA, MHC, class 1 expression, and PD-L1. If the tumor happens to mutate or get rid of the expression of JAK1 and JAK2, or one of them, there will be this pathway will stop. And that's precisely what Tony Rivas reported in several patients with acquired resistance to PD-1 blockade in melanoma. And I'm going to tell you a little bit a particular observation that we are proud of in our institution, which is using interleukin-8 as a biomarker. We recently published several papers in clinical cancer research and annals of oncology about it. The interesting thing is that IL-8 is produced mainly by malignant cells, and in the cancer patient peripheral blood, often you can detect high levels of IL-8, which is a chemokine that attracts myeloid cells, particularly neutrophils, to the tissue where it is being produced. Interestingly enough, if you xenographed mice with tumors, with human tumors, immunodeficient mice, of course, what happens is that the human tumor produces IL-8 that you can detect in the peripheral blood. But interestingly enough, this gene is not conserved in rodents in such a way that all the IL-8 you measure comes from the tumor. And as long as the tumor grows, you start detecting more and more IL-8, and if you perform a surgery and remove the tumor, the IL-8 levels drop. So you can use this as a surrogate marker for tumor volume. And this actually works as that. So the patients with heavy burden have more IL-8 than those who do not have a heavy burden. But more importantly, what we found is that patients who were treated with PD-1 blocking agents, if they eventually responded, we see a decrease in IL levels as opposed to the peripheral blood samples of those patients who do not benefit from treatment and the disease progressed, and this was true in melanoma, in non-small cell lung cancer, also in hepatocellular carcinoma. Interestingly enough, if the parameter that you measure is not the absolute value of the concentration, but instead of that the change, whether it goes up or goes down, then the predictive value becomes quite reliable. And although these series of patients are not that large, this is being prospectively validated in large serum collections. And of course, this is not a predictive biomarker because you assess it 3 weeks after starting treatment. But don't forget that we very badly need this kind of biomarkers early on treatment so we can save cost, we can save side effects, and also we can save time in order to get another line of treatment to the patient early on if we already can define the patient early on and probably go to another clinical trial or whatever other therapeutic option. Another interesting thing is that in immunotherapy, as you have learned in previous talks, it's treatment modality that can result in what we call pseudoprogression due to tumor inflammation that by imaging techniques is wrongly interpreted as a tumor progression, while what is really happening is an immune response. This is not that frequent, but often is the case that you doubt on the first attack, on the first CT scan, that the patient is not responding. So in this case, what we found is that IL-8 seems to be good at telling apart those cases of true progression versus pseudo-progression, as happened, for example, in this case of a patient with bladder cancer treated with atezolizumab. Again, this correlates with survival following treatment. So, to sum up, antigenicity, immunogenicity by proper antigen-presenting cells, presence of mechanism of immune escape in the tumor, presence of T-cell infiltrates, pre-treatment, and immune signature, particularly those related to interferon gamma expression at the tumor tissue, are supposed to be the main ones we are going to rely on from a practical point of view. Bear in mind that PD-L1 determination in tumor tissue as a single parameter is not good enough, although it's helpful and probably will let us stratify patients very well in clinical trials. And certainly we are going to go to these multi-parameter type of scores, including the tumor PD-L1 status, but not only. And probably we will have to go to more quantitative measures rather than just the expression, the number or the proportion of tumor cells expressing PD-L1 on the surface. So now we are going to switch gears and we are going to speak about immunotherapy of renal cell carcinoma. As you know, this is a terrible malignancy. It's very often diagnosed in an advanced stage, and it's a tumor which is immensely vascularized. It's a tumor with the hypoxia signal on most of the time, and it has many vessels in its microenvironment. It is a very interesting tumor because in the previous part of my talk, I mentioned to you that there was a tendency in the sense that those tumors which have an important number of T lymphocytes, pre-treatment on them, tend to be those which respond to immunotherapy and those with a better prognosis. Actually, in some tumors like colon cancer or ovarian cancer, this is an incredibly good prognostic factor. But in renal cell carcinoma, for whatever reason that we don't know, things are actually the other way around. Those tumors with a kind of denser T-cell infiltrate of T-cells producing interferon gamma fare worse in terms of prognosis. We don't know that. We don't know the reason for that, but it's something that will make this tumor kind of peculiar and interesting with regard to immunotherapy. However, When the first antibody against PD-1 got into development in a landmark phase 1 clinical trial, nivolumab, disclosed its very first data, it became very clear that this was a tumor that was amenable to immunotherapy, with an important number of patients developing responses to treatment in the different dose level cohorts of these phase 1b clinical trials. And importantly, responses, when they did occur, tended to be durable. In a way, this was one of the tumors that was selected together with melanoma, metastatic melanoma, and with non-small cell lung cancer, because there was previous evidence of a response to immunotherapy. Actually, treatment with high-dose IL-2 and with interferon had shown signs of clinical activity and even gave rise to approvals in this disease setting. Prompted by these results, there was a phase 2 trial, was a randomized phase 2 trial testing 3 different dose levels in a randomized fashion, and patients were treated until progression of or intolerable toxicity. I will tell you later why that's important. And then there were very strong signs of long survival, medians of survival beyond 2 years, and in the 3 dose levels. But I must tell you that in these 3 dose levels, probably we are achieving full receptor occupancy, at least in peripheral blood. So these data were really, really encouraging in second-line metastatic renal cell carcinoma. Hence, a phase 3 pivotal trial was designed by the same company and was launched testing nivolumab in second-line treatment of metastatic renal cell carcinoma in comparison to the most often used second-line treatment, which is the mTOR inhibitor everolimus. And the primary endpoint was overall survival. Interestingly enough, there was a good number of patients, going up to 25% of these patients, who experienced responses by RECIST 1.1 criteria, which was really encouraging. Actually, this clinical trial was halted by the disease monitoring committee because of superiority of treatment. And actually, the responses, when they occur, tended to be durable, with many of them ongoing at the time of analysis, and clearly superior to everolimus. The curves, the Kaplan-Meier curves depicting survival were very interesting for second line, although I must say that we were surprised by not seeing a plateau of patients, of the curve, eventually talking about a very long-term patients benefiting from treatment. Nonetheless, this is a clear positive phase 3 clinical trial. Although, importantly, it would have been missed if progression-free survival would have been used as the primary endpoint, since no difference was seen there. So a lot of patients were developing stable disease, and that was probably part of the reason of the extended survival. From the point of view of the waterfall plots, you can see that this is really interesting for patients with metastatic renal cell carcinoma with a slow progression or tumor shrinkages as the best response that were not that would be underestimated by the overall response rate. In fact, another very important observation was made is that some of the patients were treated beyond progression and some of them were not. And when you dichotomize for these patients, it looks like treatment with PD-1 blockade beyond the point of time when radiology progression was called, benefited the patients, as you can see in this comparison. From the point of view of safety, there is nothing really to add to what you have already seen in other presentations of immunotherapy. And in fact, probably there was a better tolerability in many of these patients, perhaps with pneumonitis being the side effect that we are more fearsome in the cases that were treated in this pivotal trial with more than 400 patients treated in the nivolumab arm. So following this pivotal trial, this is the scenario of how we guide treatment of metastatic or locally advanced renal cell carcinoma that is not surgically amenable. First line of treatment has several options, including, importantly, clinical trials, and more importantly so, clinical trials of immunotherapy. But now we have sufficient degree of evidence to indicate nivolumab as a second-line treatment. We don't know yet if it's better to use it in any other disease setting, but certainly it's useful in second-line treatment of renal cell carcinoma. Atezolizumab, as a PD-L1 blocker, has already been tested in phase 1/2 clinical trials for renal cell carcinoma. As you can see here, there is activity going to a nice response rate that, interestingly enough, seems to not to correlate with the real degree of PD-L1 expression in the tumor. It's a very, very, very small correlation. Importantly, patients treated tend to survive very much. I mean, these curves for second-line renal cell carcinoma are truly interesting, although we don't have comparative clinical trials yet. Immunotherapy with anti-CTLA-4 antibodies, in this case with ipilimumab, has also signs of clinical activity, and there are cases who responded to this CTLA-4 blockade, although they were a minority. However, this was the clue to start combining this drug in several settings. In fact, ipilimumab is being combined with nivolumab in a multi-arm clinical trial, which is extremely interesting. And even in this kind of phase 1, phase 2 setting, it's clear that seeing responses that go up to 40% of the patients is nothing that has been seen before in the treatment of second-line metastatic renal cell carcinoma. Another thing to consider is that vascular endothelial growth factor, that many of us have studied very well as the main driver of tumor angiogenesis and vasculogenesis, is probably also a very important immune suppressor. There is plenty of evidence that abundant VEGF presence in the tumor microenvironment actually decreases the level of immune cell infiltrates in the tumor and their function. In early observation, it was found that atezolizumab plus the VEGF blocking antibody bevacizumab has activity that looks promising and potentially giving rise to synergy in metastatic renal cell carcinoma. And in this trial, it was very interesting to see that when vascular endothelial growth factor was blocked, there was a more prominent infiltration of T cells in the tumor with higher levels of expression of MHC class 1 molecule and PD-L1, probably as a result of local release of interferon gamma. And thereby, it looks like there is synergistic activity when using inhibitors of the VEGF receptors, type 1 and 2, together with the different agents blocking PD-1, PD-L1, such as nivolumab, avelumab, pembrolizumab, in this case, using different compounds known to inhibit the tyrosine kinase activity of these VEGF receptors. Therefore, this is really an interesting option to be further explored in this disease. And in fact, there are many ongoing clinical trials testing immunotherapy in first-line renal cell carcinoma. Some of them are already fully accrued in such a way that we are expecting results soon, and it could be the case that some of these combinations, remember that anti-angiogenic compounds are the currently most used treatments in first-line renal cell carcinoma, but all these trials are really addressing the point of seeing whether in first-line addition of PD-1, PD-L1 blocking agents would would give rise in a comparative fashion to a better outcome. Remember, we are at the beginning. This is really the beginning of clinical exploitation of immunotherapy. We are very much in need of good biomarkers, and we certainly expect progress soon in renal cell carcinoma immunotherapy, although, as you've seen, there is a strong case for using this treatment modality in the second-line setting. I hope that during the talk I stressed enough that we are at the beginning. This is not the end, probably, and this is not even the beginning of the end. And to guide us, we are very much in need of biomarkers, particularly predictive biomarkers. And then the second message to take home is that we expect progress in renal cell carcinoma immunotherapy in the very near future, also in the direction of biomarkers as well. But as you've seen, there is a very strong case for using immunotherapy with nivolumab in the second-line setting. So thank you very much for your kind attention. And the take-home messages are that we are at the beginning of something rather than at the end of something, or even at the beginning of the end of something. And immunotherapy is here to stay, and it's in very bad need of biomarkers, particularly predictive biomarkers, as we've seen during this talk. And then an important message is that expect progress soon in renal cell carcinoma immunotherapy. It is really something that is going to come in the near future. But remember that it's already reality in the case of nivolumab for second-line treatment of renal cell carcinoma in a metastatic stage where it has a very profound effect. So thank you again for your attention, and I will leave you now with the next program, which is efficacy and safety of immune checkpoint inhibitors in non-small cell lung cancer. This has been a revolution of the PD-1, PD-L1 pathway as well. This lecture will be given by Dr. Peter Schmid. Thank you very much.